Differential gene expression, so this idea that different types of cells express different genes at different times, is what leads us to have a complex multicellular body with lots of different types of cells. And this is going to start early in embryonic development. So we start with a fertilized egg, which is a zygote, which is a single cell. And that single cell has to differentiate into all of the different types of cells in your body, and then physically go through the process of changing into those different types of cells. We can see here what would be like an embryonic stem cell, and these stem cells have the potential to become all of the different types of cells in your body. And it's going to be different gene expression in different regions and in different cells that are going to tell that cell what to be, and then direct that cell through the changes of the morphogenesis to become that actual type of cell. So one of the very first things that's going to happen is we have a single egg and sperm that are going to become fertilized into a zygote. And cytoplasmic determinants are something that come from the mother, from the egg, and they're going to influence that first kind of couple of divisions. So you can see that we have a lot of green triangles on this side, and then a lot of brown circles over here on this side. And those are arranged in a gradient. So you have lots of brown circles on the left and lots of green triangles on the right. So after this cell goes through its first division, what we're going to see is this cell has mostly brown circles, whereas this cell has mostly green triangles. And so these represent different proteins that are expressed inside of the cytoplasm. And now, because these two cells have slight differences in their cytoplasmic determinants, that's going to go ahead and tell them to start leading in different directions with gene expression. So brown circles might lead down one path, whereas green triangles lead down another path, and eventually we're going to end up with cells that are going to start expressing genes differently and then looking differently. Another source of developmental information is called induction. So once cells have kind of already decided what they're going to be, they can get induced by nearby cells to become the same type of cell. This is important because our muscles and our bones and all of our organs form in something called tissues, where all of the cells that are adjacent to each other are the same type of cell. So when we're developing our heart, we want our heart to all develop in the same place. So a single heart cell can induce the cells around it to also become heart muscle. That way you end up with one heart in the center of your thorax, instead of randomly kind of dispersed heart muscle cells all throughout the body, which would not function very effectively. So the process of cell differentiation actually begins with determination. So it's a two-step process. The first step is to decide what the cell is going to be, and this is going to happen by activation of something called a master regulatory gene. So in this case, the example is MyoD, which is a muscle cell master regulatory gene. Notice that this cell has not changed yet. So the stem cell and the myoblast cell are the same looking cell, except the myoblast now no longer has the option to become anything else besides a muscle. So it has been determined. Determined is irreversible. So once you are determined to become a particular type of cell, you have to then proceed with differentiation. So the second step is differentiation. So now that it's determined to be a muscle cell, it's going to go through the process of activating the genes that are going to activate gene expression towards becoming an actual muscle cell. And so you can see here, this is a muscle fiber. So determination is kind of like making the decision, and then cell differentiation is following through and then actually becoming that type of cell. We also have specific gene expression that is occurring during the development of the body pattern. So this is what part is going to be the head, which part is going to be the abdomen. It distinguishes the major axes of the body. And so this is going to be controlled by pattern formation cues or pattern formation genes, and it's going to give locations relative to the body axis. So once we've kind of determined that the head is this way and the tail is this way, then the genes will be expressed so that the eyes end up on the head, the wings end up on the thorax, the legs end up on the thorax, and the reproductive organs end up in the abdomen. So we want to make sure that the right type of tissues develop in the right positions in location to the body. The types of genes that control this pattern formation are called homeotic genes. And this was discovered in fruit fly experiments, where several researchers took the genes that they thought might control for pattern formation, and they looked at them in embryo stages, larval stages, as well as adult stages. And what they did was they created mutants. So they took mutations in these genes and conducted all sorts of breeding experiments to look for corresponding genes. They were able to identify many mutants that were embryonic lethals, where if that gene got mutated, then the fly never developed and it was basically lethal to the fruit fly. So that would be kind of like if a gene that went wrong with our human chromosomes led to something like a miscarriage. So that gene, what they found with these embryonic lethal genes is that they were absolutely critical for embryogenesis, and without the correct gene, the fruit fly would never develop. Then they found several genes. They found about 120 genes for normal segmentation. And so what they did is they traced these genes through embryogenesis to see what resulted when they changed these genes around or when they mutated them. One of the things that they did do is actually take genes for some segments and move them around. So you can see in this guy on the left here, the normal antega segment gene is in place, and so you end up with antennas on the head next to the eyes. In one of their mutations, what they did is they actually took a gene that coded for legs and they moved it to the place for antenna. And what happened is this fruit fly actually developed legs on its face instead of antenna. And so they were able to kind of figure out what some of these genes did by moving them around or mutating them. The axis of the body determines which end is the head and which end is the tail. And this is really important first step to kind of figuring out how we're going to position our body patterns. So we have genes that are called maternal effect genes. And these come from the female because the female has the ability to encode the cytoplasmic determinant. So these would be genes that are effective and on in the egg that then result in the formation of the cytoplasmic determinants that we think were kind of on a gradient with some over on one side and some on the other. So the first cytoplasmic determinant gene we found was the bicoid gene. And we found that the bicoid gene in fruit flies is going to affect the front half of the body. So it's going to establish the location of the head. So when they did mutation studies, they found that embryos who mother, so if the egg had no bicoid gene, so this was not available, then the embryo would end up with no head. So here would be one that has a functional bicoid gene where we have a head end and a tail end. And the bicoid gene is going to code for kind of these first thoracic and abdominal sections. This is the mutant where the mother had no bicoid gene. And what you see here is you end up just getting two copies of the back half. So you've got A6, 7, and 8 over here. Abdominal regions A6, 7, and 8 over here. So because there was no bicoid gene, the cytoplasmic determinants that allow for the development of the head, thoracic, and abdomen structures never developed. And so you end up with this mutant larva. This would be another lethal mutation because a larva is not going to survive with two tails and no head. So this was kind of the first step into this pattern development research where we were able to identify a specific gene for axis establishment of head axis to tail axis. And this was really important discovery because it was kind of laying some of the early steps of how specific proteins could be required for pattern formation. It's starting to increase the understanding of how the mother's role is super important in embryo development. The sperm doesn't bring a lot of stuff with it. The sperm really just brings its DNA. However, the mother has an egg, which lots of cytoplasm and organelles, and so the mother has a lot of potential for affecting the development of the embryo by having just more available tools. And it demonstrated that the gradient of molecules in the cytoplasm, these determinants, can determine the polarity and position of the embryo, where if we changed the gradient of the bicoid protein, we would end up affecting the shape and the head position and tail position of these larvae.